Okay, good morning. Let's have a class. And we are going to move on to the lecture 2 slides and talk about microscope and stain technology. Before that, we still do the review. Okay, and every time, like I said, you walk into the classroom, we're going to do a brief review what we talked last time. So in the slides 1, we talked about the history of microbiology. So we talk about history. We mentioned about Edward Jenner, which is the first person find and created the vaccination, which is a concave smallpox. We mentioned Anthony van Leeuwenhoek, who is the first person created a mechanic microscope. And this mechanic microscope, we said, is magnification is about 200x. And he's the first person recorded his observation of the microscope. Then we mentioned about a very important couple of research during the history about this credit spontaneous generation. And we talk about Francisco Redi, and his very important study using the maggot and the decayed beef. And we also talk about Ferdinand Kohn. They have back and forth find the microorganism from the microbiology level. But finally, it is Louis Pasteur have his very magic swan neck flask, and which is discredited spontaneous generation from the microbiology level. Then we talk about is Bob Koch. He had a lot of the contribution. A very important one, which is we talk about the Koch postulation, which is talk about the relationship between a bacteria and the microorganism and their related disease. And you also talk about the using agar as a material to cultivation bacteria, then created a concept we call it a peel culture. And we also named Fanny Hesse, Richard Petrie, all those scientists, which is gave a big contribution for the development of the agar and the peel culture. The last we mentioned about the binomial system of bacteria. And we mentioned most of that. The genome name and the species name is italic, but the exception is Salmonella. It's a difference because Salmonella we are talking about is a subspecies, it's not a species. So these are the things I want you to just go over your head what we talked, and then we move on. Now, we briefly opened the topic of the lecture 2, the microscope and stain technology. We mentioned about light microscopes. What are the categories and electron microscopes? We talk about the resolution power. We gave you the example of the light and electron microscope. We also talk about briefly in the lecture about bright field microscope because we did a lot in the lab already. We have 3 more labs talk about staining technology and using microscope. And we also talk about oil immersion to remove the air gap. So these we just briefly mention here because we're going to do more in the lab. So let's move on to today's lecture. So first thing first, we talk about the light field microscope. And there is also the one called the dark field microscope. So what is that? said very easily, light field microscope. Sorry, we should say light bright field. Not light field, light bright field. The background is bright, and your specimen, your sample, the 930 lab, you already did the simple stain. You showed up. What means dark field? Dark field is opposite. The background is dark. And then your specimen showed up. Looks like a picture there. The background is dark. And then your sample showed up the color. Now why they can do like this? This is, we need to go back to talk a little bit about the principle. So we had a microscope objective lens. Okay, this is, everybody knows, objective lens. Then we have a stage. Okay, then we have, right here, is the energy resource, we call it a light. You turn it on. Now this is a general, very common light bright field microscope. What is special for the dark field microscope? There is a block underneath the condenser, underneath the stage somehow. It's a block. It's a solid block. Once the solid block's put it there, the light going through here, it's go this way, then go this way. It is 90-degree angle. We call it perpendicular. Because of that solid block, the background is dark and the specimen is showed up, is color. Now why this is important? Because in the clinical area, there are very special type of the bacteria called spiral sheet. Spiral sheet is somehow like this. It's very soft. We call it an asexual filament. And they also could move. So they have an endoflagella. They could move it. These type of the bacteria, because of the like soft asexual filament, you cannot use traditional stain technology, like a gram stain, to do the observation. But in the clinical area, it is important. Why? There are three important categories. Three big examples. Number one, Treponema pallidum. This is a pathogen of spiral sheet cause the number one sexual transmitted disease in the world, called syphilis. That's the number one sexual transmitted disease before antibiotics created. That's caused a big damage, especially in the Asian countries. Sexual activity person were easily to get it. That's syphilis. Second one, Borrelia burgdorferi. That's Lyme disease. Some of you heard about it. What's a Lyme disease looks like? In the arm or the leg, you have these target rash or red circle. That's called Borrelia burgdorferi. Last one, leptospira interrogans. What it looks like? It's a hook shape. This is a pathogen. Is usually will be transmitted to the rabies, skunk, mice, it will cause the liver damage. And the eye, you will see like a yellow, drawn lines. These really passages, the spiral sheets, is very important in the clinical area. We cannot use a traditional staining technology to do the observation, so we need a dark field microscope. That's what it is, why it is important. So the slides here, which has showed you briefly the knowledge behind that and the principles behind that. Also the comparison between bright field and dark field. This is a good example of treponemia pallidum. You can see asexual filaments, looks like zigzag, and the background is dark. It showed up. It's a pathogen called syphilis. We will mention again in the exam 2 when we move to human infectious disease, when we have a section talk about sexual transmitted disease. Okay, so that's dark field microscope. Next one, phase contrast microscope. So phase contrast microscope. This guy is a little bit of physics behind it, but I will try to make it simple. So it's a light microscope. Light microscope, they are related to light wavelengths. So for example, this is energy resource. This is the view area, okay. Let's say general light microscope, what it looks like. The wavelength looks like this. Okay, I have another wavelength, like this. Okay. So, a very basic physics behind it. If you see two peaks there, it becomes more lighter. You see the bottom of a light, it's darker. It's contradicted with each other. It's a traditional one. Okay, just a general light microscope like the one using our lab. What is phase contrast? Somehow, around here, they put a ring. We call it a phase ring. The phase ring could be different size or different category. Once the phase ring put it there, what happened? The light wavelength will be changed. It could be either 1/4 wavelength lagging behind, we call it out of phase. Actually, it's lagging behind, out of phase. Or it could be a half wavelength ahead. So, which means it's going to change, is that right? For example, your overlap with each other becomes lighter. This curve could become like this. It could be like that. So originally, if the curve like this, you're not going to see anything. But when the phase ring changed, it showed up. You can see something. So that's a basic principle behind the phase contrast microscope. So wavelength, the speed is changed. Then will be showed up in the screen. So why it is important? We wanted to see the internal structure of some of the microorganisms, especially the eukaryotes. Some of the microorganisms between fungi and the bacteria. Let's say we mentioned about the slime mold. Sometimes it's a protozoa or lower developed protozoa. We want to see the internal structure, but does not mean every lab or every place has an electron microscope, because electron microscope is expensive. So some of the times we're using phase contrast microscope. You can see the internal structure. You can see right in the middle. This is a bright field microscope like the one in the lab. We barely can see something. Right here, you can see very obviously internal there some. Yeah, So for example, if you get the tissue or the organ or some culture or sputum sample from the patient, when you see these things, you know that the bacteria or some fungi or some of the eukaryotes has been hosted in the patient. So that's a reason why we use phase contrast microscope. This is dark field microscope. So obviously you can see, if you want to see the internal structure, phase contrast microscope is better. And here the example is the Saccharomyces cerevisiae. This is exactly the one what you guys did in the first lab when we practiced using microscope. That's a Saccharomyces cerevisiae, the body yeast. Okay. So that's the story with phase contrast microscope. Okay, fluorescence microscope. We have a couple of slides talk about that. Okay. So next one, the fluorescence microscope. Fluorescence microscope. First of all, fluorescence is a light, shiny yellow color. Some of the bacteria naturally has fluorescence. A very good example is Pseudomonas fluorescens. Pseudomonas is a sporage bacteria, widely existing in the food if you keep in the refrigerator for a longer time. So if you buy a fish, you get it from Kruger, and then you put on your dorm in the refrigerator, you forgot to cook it. About like 10 days later, you smell it's really bad. Of course, ammonia going to generate it. At the same time, if you do the analysis, you see the huge amount of the Pseudomonas. And the Pseudomonas has a light shining color, light shining yellow color, it's a fluorescence. So some of the bacteria naturally have it. What it means by fluorescence? What is the difference between this microscope with light microscope is on the top of the sentence. This is using the energy resource, relatively different, is using UV light, fluorescence light, or blue light. What this means? This means it gives you a very much shorter wavelength. So remember the RP equals lambda divided by 2 NA. I said this guy has to be the smaller the better. Because the smaller, which means the resolution power is small, and the resolution capability is increased. This is much shorter wavelength. So the resolution power is increased dramatically. This is the first thing. And plus, you will see there is a couple blocks in there. We call it a dichromatic mirror. Some of the light will be passing through, some of the light will be refracted, which means it's more focused. So that's the first thing for the fluorescence microscope. Second thing, fluorescence microscope, we have to do the staining. But the stain is different from what we did in the lab for simple stain, gram stain. We need to use a very special kit. And here gives you some of the examples. Usually in the lab, we do a lot, is using Davy stain. It's a commercial kit name, Davy stain. Why we use Davy stain? Because Davy stain is staining DNA. Because it's staining the DNA, refracted with a fluorescence microscope will show you two different colors. Is green color indicate live cell, and red color indicate dead cell. So that's a very good advantage. Using fluorescence microscope, you can differentiate live cell and dead cell. That's a good example. You can see the picture there. There's a live cell, is green color. The dead cell is red color. Right side you see the fluorescence, the shiny one, that's streptococcus. It's very long cock cell. It looks like it's injured. At the bottom is a pseudomonas, it's a eukaryotes. Some of the fungi coming from the aquatic environments. So pretty wide range of the application. You could see the bacteria, could see the eukaryotes, and you could differentiate dead and live cell. Next one, laser confocal microscope. This one, I have been using this in the research when I graduated from my PhD degree and worked briefly at USDA ALS in Beltsville, Maryland. The place is going to be going to be taken away because the new policy is that those scientists will move to Colorado, Utah, all those places, or North Carolina, which get close to the farmers. I used to work there, and I did the research using the laser confocal microscope. So, laser confocal microscope. So, what is that? First of all, laser confocal microscope is expensive. It's very expensive, somehow between 50 to maybe $150,000. Could be up to $200,000 somewhere. It's expensive. The reason why it is expensive is the laser. Lots of time we call it an argon laser. And it's coming from a very special company called ZES. That's a company generating these argon lasers. And these lasers, why we using that? Is back to here. The wavelength is also much shorter. So much more better resolution power. Laser confocal microscope, you also need to do kind of Debye stain, which means staining the DNA can differentiate live cell and dead cell. Same thing. The green color is live cell, the red color is a dead cell. But another good thing is that they could generate 3D images, which is connected to the computer. Now, why 3D images is important? Because laser confocal microscope is using to testing a very special bacteria community. We call it bacteria biofilm. This is a matrix of a cluster of a bacterial cell. So when will happen? It's very common in the food processing plants. So let's say today I'm processing the beef. Okay, I have a carcass here. Come here, I cut, I ground it, and then make a beef steaks or beef patties, like you eat hamburger. After we processing, on the processing bench, we have lots of the residues, like the dust, like the residue meat there. But all those meat basically carry on microorganisms. For example, fluorescence. For example, generic E. coli. Then one day I forgot to do the cleaning. I forgot to do the sanitizing, which means I did not follow the sanitation standard operation procedure. Then what happened? Those bacteria on the bench, especially on those very flat surface, will be accumulated. Then accumulated become a cluster, become a matrix, like the film of a bacteria. It's a bacterial film. Now what's the problem for these things? This will stay there forever. And the bacterial cell can also dislodge and it goes everywhere. Another bad thing is that these type of the matrix are resistant to the antimicrobials. So for example, a general cell, we call it a vegetative cell, vegetative bacterial cell. I'm using 50 parts per million, 50 ppm chlorine will kill it. If the biofilm, I may use 1,000 ppm of the chlorine, it can still stay there. That gives you a big trouble for the meat industry. So, people can using laser confocal microscope to do the research about the characteristics of the biofilm. and how to well control it. Because there is a layer, we can see the widest, the length and the thickness it is, how thick it is. The reason is laser confocal microscope can generate 3D images. That's a good thing for that. I just show you something what we did in the lab before. We don't do 3D images by the time, but we are using the color spectrum. We can see the color change. So this is the one, the color is control. You see lots of red cell. We using a combined culture of Pseudomonas and Salmonella. Because like I just mentioned, Pseudomonas is a sporage bacteria, very easy to form biofilm. So you can see the color spectrum here. On the right side, the x-axis is green color. The second treatment is we are treat the biofilm with just water wash. And you see there the red cell comes out, which means it starts to dying. And the color spectrometer, you can see, x-axis is still green, y-axis start to show some of the red. This is the one I'm using the chlorine. I'm using 50 parts ppm chlorine. You see the cell is dead. So the color spectrometer start to show up is red. However, still lots of the green color, lots of live cell. Like I just mentioned, using 50 parts per million chlorine is not enough to kill the biofilm. The last one is we're using a surfactant. It's called polyglycolin. It's a surfactant which one of the chlorine could be more effective to the bacteria, which means the bacteria could be more exposed to the antimicrobials. Then you see it's a purple color, which means they're injured. And you can see the color spectrometer is right in the middle. It's like a 40, 60 degree straight line curve there. So this is the example using laser confocal microscope. From 2D images, but with color spectrometer, you can see the results when they treated with chlorine solution, what it should be looks like. Okay, so just give you some of the examples. Okay, next one, we want to talk about spending a time, talk about the staining technology. This will be closely corresponding to what you learned in the lab sections. So we'll talk a little detail here and something we will be talk a little briefly if we'll do a lot on the lab. So it depends. So first of all, I want to show you. This is the process for the smear preparation, which is an example of simple stain. NICEAD lab already did. Look at there, the picture said, courtesy of Carl Zeiss microbial imaging. That's a company created and generated laser confocal microscope. So all those laser confocal microscope, we all call it a Zeiss argon laser. It's a big company, generates those products. So smear preparation, it is very simple process. Encoding on the glass slides, we draw the circle, we make a big smear. There are two things important, yes, air dry, followed by heat fixing. Okay, and then we're gonna add dye there to staining 60 seconds, and finally we are going to do a rinse. Now obviously we usually do 10x, find the specimen, and moving to 100x with oil immersion, we will find the morphology and also the arrangement. So we want to talk a little bit about the dye here. There are basically two different types of the staining. The one what we did in the lab, we call simple stain. We are using crystal violet, using safranin, using malachite green. That is we're staining the bacteria. Okay, background is bright. Why we can do that? Because the bacteria grow 37 degrees Celsius 24 hours, is positive, negatively charged, and these die are positively charged. That's what we can do. It's a simple stain, and we also call it a positive stain. But lots of the time, we have to do is negative stain. Negative stain is using Indian ink and nigrosin. We are not staining the bacteria, we are staining the background, and the bacteria show up. A very good example is this guy. You can see the picture here. The background is completely dark, and in the center, you see here, it showed up. And I can draw like this. See, it showed up. And this is very good to using negative stain. The white smear there, the white color there. We'll mention next week, this is capsule. Because bacteria has capsules, it could be concave, the self-defense of the host. Go inside, you see these? This is a typical diplococcal. Which bacteria is this? That's Streptococcus pneumoniae. This is the first pathogen we'll talk in the exam 2. The pathogen cause community-acquired pneumonia in the United States, the number 1. Okay? That's why we're using negative stain. So here we just talk about the simple stain. The dye is positive, the bacteria is negative. Negative stain, we use Indian ink, nigrosin, or ethane, or rose bagel, all those things. Those are staining the background because the bacteria is also negatively charged. They are acid dyes, they are negatively charged. This is the one you already been did in the lab already, in the first, the 930 lab. So you're using the basic dye, you can see the bacteria morphology and also the arrangement. This is a good example. The top one is using crystal violet. So a little bit of purple color and for the E. coli. And the bottom one is using methylene blue to stain in corner bacteria. We'll talk about corner bacterial diphtheria, causing diphtheria in the exam 2 in the next month. So these basically, we already mentioned, we don't talk about the size, the shape, morphology, and the arrangement of the bacteria. That's why we do the simple stain. And the negative stain, this is the one staining the background, and we can see the bacteria showed up. We know the morphology, and we know what it looks like. Now we want to spend a lot of time talk about next one. It's called the differentiate stain. Differentiate stain. Basically, we're focused here. I'm going to talk about the gram stain. Then we will be doing indosspore stain in the lab next week. Acid fast stain, flagella, and capsule stain, we will briefly mention here because I used to have microbacteriophylla in the lab. After 3 or 5 years, the pathogen is dead. So acid fast stain, we cannot do it. But indosspore stain, we can do. Flagella and capsule, we don't have that capacity. So in the lab, we will do gram stain, indosspore stain. Here, we're going to introduce you all. We're going to spend here lots of time talk about the gram stain. So here is what it is. So gram stain. First of all, gram stain created by around 80 somehow around the AD 60s, Dennis Graham. It is a differentiated stain. It is a differentiated stain technology which categorizes all bacteria into two groups, which is gram positive and gram negative. Gram positive cell, it is purple color, and gram negative is pink or red color. This is special for bacteria. For fungi, we do not say gram positive or gram negative. We just say gram stain blue. Okay, now next one. What are the major steps of the gram stain? The major steps of the gram stain on the slides have 4 steps. Now, of course, you have to follow the smear preparation, air dry, and heat fixing. But when you move on to the stage 4, you have to strictly follow up the 4 steps here. Number 1, adding crystal violet, staining 60 seconds. Crystal violet is a basic dye, which means you just put a color there, put purple color there. Second step, of course you have to rinse, okay? Add gram iodine. Gram iodine also staining 60 seconds. We call it a mordant. This will make crystal violet staining stronger. The third step is important. Using 95% alcohol. Only 10 seconds. This process called decoloration. All these dyes will be dissolved in 95% alcohol. You know that. The same thing. When you're using cosmetic material for your face or your nail, if you cannot remove it, or you used up all your nail remover, you can use 95% alcohol. If you don't have it, you use some vodka. It just is the same thing. It's 50-60% alcohol is the same thing. That's decoloration. The dye can go. The last step is using saffron. This is also 60 seconds, followed by a rinse. This will be generate a pink color if it is gram-negative bacteria. So this step, we call it a counter stain. Because after staining, after the decoloration, it's blank. So we have to put something there. So that's called counter stain. Crystal violet, followed by gram iodine, 95% alcohol, decoloration, and the counter stain with saffroning. That's the 4 major steps of the gram stain. And you should start today to remember them. We're going to do a couple of times in the lab during the AEM 341 lab sections. Okay, so you should remember them. And we're going to practice next week after Labor Day. Now the question will be, why the color looks like that? Gram-positive bacteria staining purple color, it is actually crystal violet color. And the gram-negative stays pink and red. It is a counterstain color. It is saffron in color. Saffron in color. These two things you need to remember. Okay, now we talk about the theory. So why? And why gram-positive bacteria stains purple and the gram-negative bacteria stains pink or red? This is related to the cell structure. There are two theories. Most of the textbook will mention one, I'll tell you two. The first theory, called the cell wall theory. So what is the cell wall theory? You need to know, or a little bit familiar with the cell wall structure of the gram-positive and the gram-negative bacteria. We have a slide for you next week, but I'm going to draw here. So what it looks like? Let's do gram-positive bacteria. Okay, gram-positive bacteria. This everybody knows. You learned it in your biology class. Cell wall structure. Fluid mosaic model. Composed by phospholipids with protein, membrane protein, cross-membrane protein, or structured protein there, interacted with each other. That's called fluid mosaic model. That's a cell membrane system. A little bit of space here. For gram-positive bacteria, has a very heavy structure right above the cell membrane system called peptidoglycan. Very, very heavy. Peptidoglycan composed by two major chemicals, modified sugar and acetylglucosamine and acetylmuramic acids and amino acids. We're going to talk next week. Very heavy. Then on the top, relatively simple. Teichoic acid or lipoteichoic acid. That's gram-positive bacterial cell wall looks like. The key thing is here. Okay, what is gram-negative bacteria looks like? Gram-negative bacteria, same thing. The down here is the same. Okay, fluid mosaic model for cell wall. You can say it's an inner membrane system. Little bit of space here. But in contrast to gram-positive bacteria, the peptidoglycan is very, very thin. This is peptidoglycan for gram-negative bacteria. Very, very thin. Like that. Then above, you have another layer. That's called outer membrane. Somehow like this. And then you go here, you have a very toxication material will be generated. LPS. Lipopolysaccharides. So one of the major components is lipids A. That gives you a structure. Then we'll talk about this. Theory 1, cell wall theory. You can see the difference. Because gram-positive bacteria have a very heavy peptidoglycan. So, we are the crystal violet. We are the gram-iodine. Let it stay strong. Because of this very heavy peptidoglycan, we're using 95% alcohol for 10 seconds decoloration. It will not remove them. The color is still there. Then you counterstand with the safranin. Safranin, the dye is not as strong as crystal violet. So that's why it showed you purple color. So cell wall theory is very simple. Gram-positive bacteria owns heavy peptidoglycan. During decoloration process, not easily removed by 95% alcohol. Crystal violet staining stays. That's the cell wall theory. In contrast, gram-negative bacteria have a very thin peptidoglycan. It cannot hold on when you do the decoloration. And it will be this color. So it's blank then. Then you counterstain, use safranin, it showed up pink or red color. So you can say instead, gram-negative bacteria has very thin peptidoglycan. That's a cell wall theory. All the textbooks talk about that. Easily understand. And you learn in your biology 101 pretty much. The second theory I want to introduce you here is called the lipid theory. So what is lipid theory? Gram-positive bacteria basically has around 1 to 5% limited amount of the lipids. You know lipids, is that right? CH3, O, O1, then CH2, then go O2, then CH3, go here, then go R3. This could be very, very long. That's very, very, very long. That could be have a double bonds. Then you go like a cis trans. Is that lipids? Okay. We'll talk later when we go detail chemistry. So that's gram-positive bacteria, 1 to 5% of the lipids. Gram-negative bacteria has around 10 to 15% of lipids. Why? Because of the lipids A. Lipids A on the top of the lipopolysaccharides. Lipopolysaccharides is a very bad thing for gram-negative bacteria. Will generate endotoxin. This is a reason why Salmonella, E. coli, or those gram-negative bacteria is toxication pathogenic bacteria, because part of the reason is endotoxin. And endotoxin is heat liable. So you will cook it, it will kill it. It's indifferentiated to exotoxin. Exotoxin coming from Staphylococcus aureus. That heat resistant. That one you use heat, you may not kill the toxin. That's why the salad, the chicken salads, all those things have the problem all the time. If it's not well prepared, you have a diarrhea, vomiting after you eat at 3 or 6 hours. We'll talk about that later, okay? So gram-negative bacteria have a 10 to 15% of the lipids. So what happened? The key thing is still here. You add a crystal violates, you add a gram iodine to stay there. That is strong. Now you add 95% alcohol. Gram-positive bacteria have 10 to 15% of the lipids in the cell wall. So the dye there, and lipids is easily to be dissolved in the alcohol. And you know that. We draw here. Triglycerols is easily to be dissolved in the alcohol. And we call it organic reagents. So what's the definition for lipids? Because it could be dissolved in the organic reagents. Is that right? That's one of the chemistry knowledge. So lipids can be easily dissolved in 95% alcohol. Then together with the dye, so they all washed off. And in other words, gram-positive bacteria only have very limited 1 to 5% of the lipids. So it's not going to be easy to remove by the 95% alcohol. So it stays there. Plus, it has a heavy peptidoglycan. Okay, so the theory here is very simple. Gram-negative bacteria has 10 to 15% of lipids. Easily to be washed off by 95% alcohol during decarboration. Which means after decarboration, it's black, it's no color. So you can't stain saffroning showing the pink color. Okay, so these are the two things are very important when we talk about the theory behind that. Something else I want to talk about the gram stain, it is important. So gram stain, the theory, it's based on the cell wall structure. We just talked about the peptidoglycan. Which means the bacterial cell needs to have a very good or constructed peptidoglycan. So gram stain, we have to use number one, very fresh, we call young culture. What means young culture? Around 80 to 24 hours at 37 degrees Celsius, freshly cultivated bacteria. You cannot use the old one. If you are using more than 72-hour bacterial culture, what will happen? The bacterial cells started to die. The peptidoglycan already started to cracking down. You cannot hold on crystal violet. Then, even it is theoretically gram-positive bacteria, it will stay like gram-negative. It's all showed you pink color. That's called gram variable stain. So keep in mind, very easy. Has to have very fresh and young culture. The second thing, gram stain. The procedure is very important. You have to be strictly followed. But the time is not important. So crystal violet stays 60 seconds. You can stain in 45 seconds. You can stain in 2 minutes. It doesn't matter. But the only tricky thing here is this guy. The 95% alcohol decarboration, you have to restrict it to 10 seconds. If you're doing this decarboration for 60 seconds or 2 minutes, and I said a very wild example, if you're doing 95% alcohol decarboration, somebody called you to go stadium to see WVU versus University of Pittsburgh as a football. Then you go there 2 hours later, everything will wash off. So decarboration, the time 10 seconds, is important. The way we do, you either do timing or you count 10 Mississippi. So that's the way we have to control the time. Okay, that's the second thing I want to mention. Time does not matter. Procedures is important except declaration. Have to stick to 10 seconds. Last thing we want to mention, this related to the lab. Lots of the students, when they're recording gram results, they will only say it's gram positive or gram negative. But actually you need to recording 3 major items, which is gram reaction, positive or negative, cell morphology. It's a cocktail, or it's a log, or it is, or we say vibro. The last one is cell arrangement. Is that a single cell? Is that a chain, or is that a cluster, or is that a grape shape? That is called cell arrangement. Those 3 things, we will repeatedly remind you in the lab sections. It is important to keep recorded. Okay? This is very important information for the gram stand. So that simple slides right there is actually gives you lots of the information. We will practice, and we also going to talk. Okay, acid fast stand. We'll talk very briefly. Acid fast stand is using for one very special bacteria called mycobacteria. Mycobacteria has 2 major species. Is Mycobacteria tuberculosis and Mycobacteria leprae. Mycobacteria, it is very interesting. Where do you know it's existing more? Raw milk. This is a big debate in West Virginia, the center, whether should be legalized raw milk or not. People think it's a good nutrition ingredient there, but if you using high pressure cooking or you using pasteurization, you may boil it, you may kill them. But the mycobacteria is another concern. Mycobacterial tuberculosis will cause tuberculosis. Mycobacteria, other species will cause deboning, which is called hunchback. So that's a risk. This bacteria is very special because it has a high amount of unsaturated fatty acids, which means you have a double bonds. Remember I draw there? That's why I draw a brief picture. Well created. The high amount of the unsaturated fatty acids, we call it mycolic acid. The mycolic acid will make a very waxy cell wall. This is a reason we cannot use a general simple stain or gram stain to do, because of the waxy cell wall will prevent the dye penetrate. So how are we going to do the acid fast stain? We have to do is using a glass slides, a smear preparation. We're going to flooding with carbon fusion. At the same time, using Bunsen burner to do heat steaming. This is not heat fixing. It's called heat steaming for 5 minutes. We'll show you in the lab when we do the indoor sports thing. It's the same thing. Heat steaming for 5 minutes. Then the carbon fusion, a red purplish dye will penetrate. It will be penetrated through the waxy cell wall, then do the stain. And then we rinse and we counter stain with the methylene blue. And this is what it looks like. The background, the blue is methylene blue. That's called counter stain. And you should see those pink color, that is carbon fusion. Acid-fast stain in the clinical area is very important to diagnose tuberculosis. Tuberculosis in the history is like a lung cancer. Before antibiotics created, this is the one. If you get, you die. There's no anything you can do. You can go back to see lots of the musicians, lots of the scientists even, they died of, in European countries, they died from the microbacterial tuberculosis. In China, Vietnam, or those Asian countries, they have a traditional herbs. We call it Chinese medicine. They try to conquer this one, never works until antibiotics generated. Also, the diagnosis process is tough because tuberculosis is growing very slow. It's not like E. coli. It grows 24 hours. You get to 10 million cells. Tuberculosis, the generation time could be more than 2 days, which means one cell become 2 cells, took you 2 days to grow, some species. Then we have to do a diagnosis. So we're using a relatively fast way to do is using acid-fast stain. If any of the patient, when you see that from the lung area or from the lung sputum sample, this means you're suspicious of presumptive tuberculosis, which means you have to use antibiotics immediately. At the same time, the isolated species go to the reference lab through transport media to do the further confirmation. The confirmation method, right now you can use a molecular methods. It's immunology methods, which is much quicker than you growing a bacteria. But in the history, it's a really tough one. Right now the problem is more and more antibiotics using. So microbacterial tuberculosis become a multi-drug resistant tuberculosis. That causes more problem. But just want to let you know that's what the acid-fast stain what looks like. Now, why we also call it an acid-fast stain? This process, there is also a decoloration. The decoloration, we're not using 95% alcohol. We are using acid alcohol. Okay, that's why we call it an acid-fast stain. This is, I also kind of remind all of you in the lab, we sometimes have a bottle said acid alcohol. You don't use it next week for the gram stain. Because when you add acid, you add alcohol, everything will be removed. Which means everything will stay gram negative. Okay? So that's all I have for today, and we'll see you after Labor Day.